Swayam Prabha. Digital India. Educated India.
Hello everyone, this is a course on computer aided drug design. I am going to take 40 lectures, each lecture of half an hour duration. So we are going to talk about how to design drugs with the aid of computers. So computers will be an aiding tool, but you cannot replace a medicinal chemist or a synthetic organic chemist or a physical chemist in helping in designing new drugs. So this particular talk is going to introduce this topic of computer aided drug design. So what is this drug discovery? So this is a process by which a drug candidate is identified, it is partially validated for the treatment of specific disease. We are going to look at why partially validated, because unless you actually do an animal trials, unless you actually do human volunteer trials, validated compound does not become a drug which can be introduced in the market. Okay, so that is why we call it partially validated. So you need to know the mechanism of action, you need to know how the drug acts, what are the enzymes on which it goes and binds to, or a protein, thereby inactivating the particular pathway of the disease. So we need to understand the mechanism of action. So we need to identify that particular target, the enzyme or protein, and we need to validate that target. That means we need to use tools to be sure that that is the target on which my drug is acting. We need to identify a candidate. Generally, before the compound is approved by FDA and introduced into the market, it is not called a drug. It may be it is called a lead compound. That means a lead, a compound which has shown very good activity, a compound which looks very promising. So that is called the lead identification. Okay, and then we need to optimize the lead, because just because the compound acts very well in my lab does not mean it has all the properties of a drug. As you know, drug is something that is taken inside the human body, so it should not be toxic, it should not cause side effects, it should have a good absorption in the stomach and so on. So they are called drug likeness property. So we may have to modify those properties so that it not only has good activity but also has good drug likeness property. So we are going to introduce this term drug likeness property for a mini lecture. Okay, so it should have these drug likeness property. This is called ADME. That means A refers to absorption, D refers to distribution, M refers to metabolism, and E refers to excretion. Let me write down: absorption, distribution, metabolism, and excretion. That means the drug has to get absorbed into my stomach. Okay, mostly if it is an oral drug, it has to be taken, it goes to the GI tract, that means gastrointestinal tract, gets absorbed, then it gets distributed inside your bloodstream. It gets metabolized because there are many enzymes. Your liver is a gatekeeper which tries to keep on degrading any foreign compounds coming into it, and then finally it has to get excreted, that means thrown out of your body. So the drug which is taken in has to have good ADME properties, that means absorption, distribution, metabolism, and excretion properties. So it may be a very good active compound, but maybe it gets metabolized and becomes inactive inside your stomach, so then it is of no use. Okay, so ADME is also very important when you are designing drugs. Then it should have good PK and PD. What is this PK and PD? PK is nothing but pharmacokinetics and PD is nothing but pharmacodynamics. That means pharmacodynamics. That means how the drug is excreted as a function of time, how the drug is absorbed as a function of time into the plasma, that is all given by pharmacokinetics, and how the drug which is inside the body acts on the disease, that is called pharmacodynamics. We are going to talk about each one of them much more in detail, and it should not have toxicity. So, we need to know the toxicity of the particular compound which we are testing, that is the lead which we are testing. It should be non-toxic, short term toxicity as well as long term toxicity. For example, if somebody has a arthritis problem, they may have to take the anti-arthritic drug for a very long time. So, it should not have long term side effects or toxicity. Okay. So, all these properties needs to be looked at when we are designing a drug. It is not only the activity in my lab, but I need to understand what is the mechanism of action, which targets it goes and binds to, and prevents the disease progression. It should have good properties, drug likeness properties like absorption, distribution, metabolism, excretion. It should have good pharmacokinetics and pharmacodynamics. It should not have any toxicity, and so on. So, the drug discovery process does not include many steps, preclinical studies. So, I have a nice lead in my lab, so I do not introduce that immediately into the market. There needs to be something called a preclinical studies. Normally that is done in with the help of animals, maybe rats, rabbits, dogs, or monkeys, and so on. That is called preclinical studies. Then comes clinical trials where we use human volunteers. Then comes regulatory approval. You need to get approval from the regulating body like Food and Drug Administration Authority, and then it goes into sales and marketing. So, these are entire drug development process actually, but we are not going to talk about all these. Our focus is more on these rather than these. Okay. So, it is much before preclinical studies we are going to stop at. So, let us look at history of computer aided drug design. It started in 1960s. Okay. So, everybody started looking at the target, okay, how the drug, which target it went and bound to. Before those 60s, a drug was given to a patient and there was a reduction in the pain or reduction in the fever or reduction in the infection, and that is it. Nobody bothered about how the drug reacted, which targets it went and bound to. Okay. Then came the 80s where they started using high throughput screening, by which many candidates were tested for certain targets, okay, in an automated way. 10,000, 20,000 compounds were tested in one shot by pharmaceutical companies. Okay, that is called in the 80s, because it is not possible just to take one compound and keep testing one after another, which may be a very time consuming process. Then in the 80s, okay, again databases started coming. Lot of databases are created, combinatorial databases on activity of a large number of compounds, activity of compounds on different targets. They are called combinatorial libraries. Then again fast computing started coming into, computing power increased, parallel processors came. So, scientists started using different parallel processing machines for performing ligand protein docking, drug protein docking, drug target docking. Okay. Then again 90s, genome assembly, genomic based target selection. That means we try to understand what are the genomes involved in the disease process. Okay. Then in the 2000 we went into pharmacogenomics, that means combining genomics with pharmacological. So, which genes affected which pharmacological issues. So, this I would say approximately the history of computer aided drug discovery. Let us look at some names of companies which are involved in drug discovery. Of course, this is more as a overall knowledge or picture. The top 10 pharmaceutical companies, you might have heard about it. This is 2014 data. Novartis, Pfizer, Roche, Sanofi, Merck, Johnson & Johnson, GlaxoSmithKline, AstraZeneca, Gilead, Takeda. Okay. and look at their annual sales in $1 million. Okay, this is almost $47 billion, $45 billion. So if you add up all these, we are talking in terms of 200 to 300 or even $500 billion. Okay, that's a big business. So drug discovery, drug business is very, very large. These are some biopharmaceutical companies. Now many drugs are being manufactured using biological route rather than chemical route. So many biopharmaceutical companies have come selectively using biological routes, whereas now these companies which were making drugs through chemical routes have also started making some products using biological routes. So some of these companies are Amgen, Genentech, Serono, Biogen, Genzyme, and so on actually. So drug discovery, if you look at it, the global scenario, the market is very large. US is the biggest market, followed by Europe, Japan, and so on actually. Let us look at Indian companies. They are much smaller when compared to, for example, Novartis or Pfizer, maybe 1/100th size, but still they play a very important role, especially in generic. Ranbaxy, Dr. Reddy's Lab, Torrent, Cadila Health, Cadila Pharma, Lupin, Sun Pharma, Nicholas Parimal, Dabur. So all these are Indian companies, and as I said, India plays a very important role in generics. That means drugs which have come out of the patent regime, which anybody can make. Indian industries are extremely competent in making compounds in a much cheaper way. So the products become very competitive, and they are able to export to US, Europe, third world countries, and so on actually. So Indian companies are not into new drug discovery, but they are into the generics and over the counter drugs, and they are really competing globally, and their products are approved by the FDA, that means Food and Drug Administration Authority, and so on actually. Okay, so let us look at the top 10 selling drugs year 2013. Okay, it just gives you an idea of what does drugs currently, which are very, very important. Okay, so you look at this, the top selling drug in 2013 is an anti-inflammatory drug for rheumatoid arthritis, 14.8 billion sales. Okay, that's a big money. Then this is also something to do with the arthritis, autoimmune disease, manufactured by these companies, that's also very big. Then comes another third compound which is also for rheumatoid arthritis. Then we have a drug for asthma, diabetes, cancer, so several drugs for cancer, okay, and then cholesterol lowering drugs, and so on. So all these top selling drugs as of 2013, they are all in the range of 14 billion going right up to 5.5 billion sales. And though the drugs which are marked in red, okay, are manufactured using chemical routes, whereas other drugs are manufactured by biological route. So what it means is slowly the pharma companies are moving towards making drugs using bioprocess approach rather than chemical approach. When you say bioprocess approach, we are using either an animal cell or a plant cell or a bacteria or virus or a yeast cell and so on to make the product. Okay, only 3 compounds are fully manufactured using a chemical route. I think as time progresses, all these molecules may be manufactured using biological route. Okay, so the lecture which I am going to give in the next 40 lectures will cover topics like structure and property. So what are the structural features that a drug should have which will lead to certain property? Okay, for example, property could be solubility, property could be its lipophilicity, and so on actually. Even properties like toxicity, degradation, stability at different pH condition, they are all called properties. And then drug likeness, does it have the drug likeness property? That means just because I design a molecule and it shows very good activity does not mean it can be taken in by human, because like I said, it should be less toxic, it should not have side effects, it should get coming out of the body after it is done its function, it should be stable at various pH conditions. So these are all called drug likeness property. So we are going to talk about that. Then of course absorption, how is it getting absorbed into the body, distributed, metabolized, excreted. And then oral bioavailability. So we take the drug orally, so that means it goes into the stomach, where the stomach is highly acidic, we are talking in terms of pH is equal to 2, and then it gets absorbed into the stomach, okay, it goes to the blood, and then from the blood it gets circulated into the body, and the liver, which tries to degrade whatever foreign objects there, so it has to overcome that, and finally it has to reach the target site. So that is called oral bioavailability. So just because I take a 10 milligram tablet does not mean at the target site the concentration of the drug will be 10 milligram, because it might not be absorbed properly in my stomach, or it may get degraded in the acidity in my stomach, or it may get degraded by the liver enzymes, and so it may, whatever concentration that is reaching my target site could be very small. Okay. So that is called oral bioavailability. So we are going to talk about that. Then we come to force fields. What are the different force fields we use while molecular modeling, okay, and then how do we calculate minimum energy conformation, how do we minimize the structures of molecule, what are the boundary conditions we use, okay, QSAR, that is quantitative structure activity relationship, quantitative structure activity relationship. Then we are going to look at the structural features of the drug, that is called the pharmacophore, and then we are going to how to modify the structure, that is called scaffold hopping, and then we look at how targets are being identified, how do you identify how the drug goes and binds to a particular target. So we are going to look at docking, and then we are going to look at pharmacokinetics and pharmacodynamics, that is PK and PD, okay, whatever I said. So in the next 40 lectures we are going to look at all these topics. We are also going to look at certain softwares which are freely available, which we can use for performing all these operations. There are many software which are freely available, so we do not have to buy any commercial software. I am not going to talk about any commercial software, but all the freely available softwares we can use to perform several of these operations. Okay, there are very good freely available softwares in the web. We are going to look at all of them. Okay, so what are the computational resources for this? Okay, there is something called Swiss ADME. I have given these site addresses also, so you people can have a look at it. One is called the Swiss ADME, which can give when you draw a structure, it can give properties of molecules, it can give what is the oral bioavailability. Then we have the Swiss target prediction. It can predict what are the possible targets if I give a structure. Okay, that is also very useful actually. If I give if I give a molecular formula, it gives you the molecular structure. This software predicts the pharmacokinetic properties of a drug candidate using QSPR. Okay. Then this software predicts toxicity of molecules, and also toxicity of the fragments of the molecules, because when a drug goes inside the body, as I said, the enzymes in the liver breaks the drug into small fragments. So the fragments also could be toxic. This software tries to predict whether the drug itself has toxicity properties, or the fragments that are created generated by the degradation of the drug also has. Then we have the QSAR toolbox, Then we have chemi informatics, ADMAT, chemical predictions, this software called VLS 3D, okay. And then there is something called a 3D QSAR, 3-dimensional QSAR. There is a software for this. Software gives you the pharmacophore details, that means it can identify the features of the molecule and try to search millions of chemical structures, okay, which have this same pharmacophore. This another software gives you the QSAR pharmacophore model. Then we have a property prediction, Osiris. Then we have another properties prediction that is called eDragon, and there is another software for property prediction that is called Mol Inspiration. So you see a lot of softwares available, okay, and during the course I am also going to use many of these softwares, and I am going to demo to you people how to use some of them based on my experience and so on actually, okay. Then there is another software, it's called a therapeutic target. So it has data information about therapeutic protein, nucleic acid targets described in the literature, including the targeted disease condition, pathway information and so on. Then there is another software called Stitch. So it connects the chemicals or the drug with the target and genes, so it creates a very complicated map, okay. It stitches basically between the genes, the target, the drugs, and creates a very complicated map connections, so it can connect almost 1.5 million genes. Then there is a T3DB toxin target database, so the common toxins and their associated toxin targets we can identify using this database, okay. So a lot of softwares, and I am sure you guys may be finding your own software as you keep searching the web. Books, there are some good books are there, drug like properties, concept, structure, design and methods, ADME to tox, okay, this is a very good book, okay. And then you have guidebook on molecular modeling in drug design, pharmacophore perception, silico drug design, software and resources for computation. So very good books are there, you guys can have a look at them. Databases, we have the zinc database. Zinc has 35 million purchasable compound structures, okay. So if I am going to look at those structures, those properties, and I find one molecule looks very promising, I can even buy from them, okay, for a cost. So this is a very useful database. Then we have the drug bank database, okay. There are 8,200 drug entries in that, that means drugs which are approved by the Food and Drug Administration Authority, okay, that is FDA approved drugs, okay. Then biotech drugs, nutraceuticals, experimental drugs. So if I want to know if there are any drugs for a particular disease, I would look at this particular database or another database called drugs.com. This has got 24,000 prescription drugs, over the counter medicines, natural products. So first step, if I want to know whether there is any drug available for a disease, is that I will go through these two databases, okay. Then you have ChemSpider, ChemDB, Chemi Informatics tool, PubChem, ChemMBL. All these databases give structures of molecules, structures of drugs, structures of prescription drugs, or structures of nutraceuticals, okay, and some properties like solubility and lipophilicity, molecular weight, number of rotatable bonds, number of hydrogen bonds and so on actually, okay. So these are, I would call, databases very useful for if you want to look at structures. More of it. Pharmacogenomics knowledge implementation, this is another database called PharmGKB. This is a resource that curates knowledge about the impact of genetic variation on drug response, okay. So if there is a genetic variation because of different races or different communities, then how the drug will respond. So there is a lot of information, okay, data on genes, diseases, drugs, pathway. So we can look at this database also if you are interested. Then of course Medchem Designer. So large number of databases, structures of millions of molecules are available, as a resource. Okay, more of this database of interest for drug discovery. Okay, so we have of course, as you can see, some of them are commercial products. Okay, some are publicly available, freely accessible for academia, commercial version also available. So we can have large number of databases on molecules of different structures. Okay, of course, some of them are commercial also. That means if you make a payment, you will be able to get databases for you, which can be used for in silico analysis. Journals, these are some few journals that are talking about computer aided drug discovery, medicinal chemistry, drug discovery today. Some of them deal with the reviews, and some of them deal with the experimental studies and so on actually. Okay, so large number of resources I have introduced it to you. You can play around, look at some of these structured databases as well when you have time, okay, and see how useful they can be. Okay, let us look at some structures. Aspirin is a molecule we all must be knowing for a very long time. We all would have taken this particular drug. It is an analgesic, that is a pain reliever. It is antipyretic, that means it reduces fever, anti-inflammatory, that means it reduces inflammation, and anticoagulant. Aspirin has become a wonder drug. Nowadays it is being used if somebody has a problem with the cardiovascular problem, and if the blood viscosity has to be reduced, they are asked to take aspirin. So that is also used as an anticoagulant. So it inhibits the production of prostaglandins. Okay, these prostaglandins are mediators which carry pain. Okay, this is the structure of aspirin. Is the acetyl salicylic acid. Okay, acetyl, this is the acetyl group. Okay, this is the salicylic acid. There are many variations of aspirin, because you can think about different substitutions and you can have different structural features. So if you want to go into a drug discovery, we are talking in terms of 10 to 15 years. It costs about $1 billion. You may have to test maybe 10,000 compounds, animal trials or clinical trials or in the lab, and then you may have to test it out on about 1,000 to 5,000 volunteers, and hopefully you may have one successful candidate. Okay, so drug discovery is like a lottery. In computers we may have to test maybe 100,000 compounds, in the lab we may have to test about 5,000 to 10,000, and finally you may succeed with one drug, or some of them, most of them will fail. So a lot of money is lost because of this particular exercise. That is why when drugs are introduced, a newly introduced drug is always very, very expensive, because there is a lot of money involved and there are many failures for every drug introduced into the market. Okay, so in the 60s drug discovery used to be cheaper, talking in terms of $4 million US, but as you can see, as you go down, it has become almost $1 billion. Okay, simultaneously in 50s maybe only few compounds were tested, but as you go down we can see we are talking in terms of 10 times more compounds are being tested. So we started testing more compounds. The cost of marketing a drug also keeps going up and up and up. Okay, and there are only 6,000 known drugs, but there are 10 power 60 possible compounds. So obviously there are many drugs in this compound list which we do not know what type of activity it will have. Okay, so there is a lot of scope for developing new molecules and new compounds and so on. So we will continue in the next class of the concept of drug discovery and what are the steps involved in target identification and lead identification. Okay, thank you very much for your time.